Good morning student. Today we are going to do the protein test and that we are going to do the Biuret and the Xanthoprotic test. But before proceeding for the test, we should know what is a protein. What are proteins? They are complex specialized molecule and they are composed of carbon, oxygen, hydrogen, nitrogen and sometimes they contain sulfur also. Now these proteins, they are the building blocks of our body. Why? Because they contain amino acids and these amino acids, what they form? They combine to form polypeptides. That is proteins. The different amino acid, they have different side chain. So the proteins, they have many important role in organisms. They are structural proteins. Example of structural proteins are collagen and elastin and they provide support. Then there are some regulatory proteins. Can you name regulatory proteins such as enzymes and they control the process. And proteins, they also play an important part in the immune system. What are they doing in the immune system? Antibodies and oxygen transport, that is through hemoglobin and the movement of the muscles, etc. It is done with the help of the proteins. So you can see proteins, they are so important constituents. But you have students, you have observed that when we are adding a reagent to a particular solution, then we get color. Why do we get a different color? Characteristic color. This is due to the formation of a complex molecule. A complex molecule is formed when some chemical reagent is added in the test. So similar here you will be observing when we are doing the test for Biuret and Xanthoprotic, then you will have the characteristic color. So coming to the Biuret test, what are the objective? What is objective? We have to detect the presence of proteins in the given solution and it is a general test for protein. What is the principle? Generally when we are doing, we have to write the principle. Then Biuret test, first you know it is a general test for proteins and it gives color reaction. Why it is giving? Because of the presence of peptide linkage in polypeptide or protein. These peptide linkage, they are present in the protein and they are forming a long chain that is of polypeptide. Since all proteins, they contain peptide linkage. Every protein has a peptide linkage. Secondly, the compounds, they contain two or more peptide bonds. And when these bonds, on these bonds, this reagent is acting and it is giving the characteristic color. And the name of the test is given from the compound Biuret and the structure of the Biuret, what it is made up of two molecules of urea and when they are heated, the exact reason of the color is not known, but it is that when they are heated, then they are giving a color. According to one common view, the color is due to the coordination of the complex of the copper atom with 4 nitrogen atoms of the protein. 4 nitrogen because it is made up of two molecules of urea. So what are the reagents we are using? Reagent in Biuret, it is sodium hydroxide 10% solution in water and second is the copper sulfate, that is 0.5% solution in water. So the percentage of the solution will be, what will be the 10% solution of sodium hydroxide and 0.5% solution of copper sulfate. Now what is the procedure? How we are going to do the test? So before proceeding for the test, we require the apparatus. What the apparatus required? We require a test tube stand, we require test tube, we require prongs, dropper and the reagents for the proceeding the exam. So how we are going to do? We will take two test tube. One test tube will label as test and another as B. In both the test tube, what we are going to do? We are going to add 2 ml of the protein solution in test tube T. But in test tube B, what we are going to add? 2 ml of distilled water. We are taking two test tube. We are marking as T test and another is blank B. In the test tube T, we are adding 2 ml of the protein solution. But in another test tube, we are adding 2 ml of distilled water. Now what we do? We add 2 ml of sodium hydroxide in both the test tube and mix. And after that, we add copper sulfate solution drop by drop. Drop by drop we are going to add, and what we see, what the observation we see a purple color, it appears the presence of protein in T test tube. T test tube we will see the violet color, but in B test tube it will be blue. So generally this purple color is due to the presence of protein, and sometimes peptones they give pink color and require large amount of copper sulfate solution. If suppose pink color is coming, then you add few drops of copper sulfate, and then sometimes gelatin it is present, so it gives a faint color. Now you can see in the diagram very well it is marked how that we have taken the sample and then we added NaOH. After that we add drop by drop copper sulfate solution, we are shaking, and then we are getting a violet color, which is a conformity test of the proteins. So this is confirmed that protein is present in the solution. Now after this we have some precautions we have to take when we are doing the experiment. So first, you should avoid excess use of copper sulfate solution. We should always use copper sulfate solution we are putting drop by drop. Otherwise, if you are putting in excess amount, then what happens? The violet color is suppressed and the solution becomes whole blue due to the presence of cupric oxide. Cupric oxide will mask the effect of violet color. And second thing you remember that magnesium sulfate and ammonium sulfate, they interfere with this test. So, if we are doing the test, we should be very well clear that the solution does not contain these two salts, that they should not contain magnesium sulfate and the ammonium sulfate. Now coming to another test, Xanthoprotic test, this is also done for protein. Now what is the principle of Xanthoprotic test? This is based upon the principle of nitrification. In this, the active aromatic amino acid, they undergo nitrification in the presence of concentrated nitric acid. जो aromatic amino acid है, उनका nitrification होगा presence of concentrated HNO3 and forms a yellow colored nitro derivative. In this, we are getting the yellow color. In the biuret we were having the violet color, but in xanthoprotic we are getting a yellow color. This is due to nitro derivative. Now what are the objective? The objective of xanthoprotic test is we have to detect the aromatic amino acid. What are the aromatic amino acid? Generally tyrosine and tryptophan. But the third aromatic amino acid, phenylalanine, it does not respond to this test. Means this doesn't give, xanthoprotic test will not come for phenylalanine. It will only come for tyrosine and tryptophan if these aromatic amino acids are present. Now reagents, what are the reagents? Concentrated HNO3 and sodium hydroxide. Here we are making 40% solution in water. Now what's the procedure? How we are going to do? We will take the test tube, and in the test tube what we are going to do? We add 2 ml of the solution, the protein solution, and then we add 1 ml of concentrated nitric acid HNO3 and mix it. After putting this concentrated nitric acid, you get a white precipitate. Safed white precipitate will come. Now you heat the solution for 1 minute and then cool in the tap water. What we are doing? Heating and then we cool. After cooling, what you see, some precipitate will redissolve and the entire solution becomes yellow. And to this yellow solution, if we add a few drops of NaOH, 40% NaOH, then the color changes to orange. This is a conformity test that tryptophan and tyrosine, it's present. Sometimes, as I told you, gelatin, what precautions, it also present, but it does not contain tryptophan and tyrosine, so it gives only a faint color. So these are the 2 protein tests, and you can see here very well in the diagram how this xanthoprotic test, you see a yellow colored solution, it is coming. This is a detection, this is a positive test used for the detection of aromatic amino acid. Hope you have understood, and till then, goodbye. Thank you.